start recording. So happy Monday. It's a holiday for some and not all, but we are having lecture today. So fall 2020 Columbus Day, here we are talking about the rest or the final part of our specific immune defenses, all right? And so we left off on our last lecture talking about, all right, we had those 7 critical non-specific immunity defenses that you have to learn. You have to learn what they are and how they work. And kind of this, I think, graphic puts it together really well, how those non-specific defenses do kind of push out in that they are our first and second line of defenses. But if a pathogen is able to evade, if a pathogen is able to propagate and multiply and spread out faster than the non-specific defenses are capable of slowing them down and removing them, then we need a way to more specifically find, target, and really ramp up a defense against that pathogen. And that's where the specific or the third line of defense comes into play. Now, we learned really, really large amounts of this information in the 70s and the 80s. And again, it comes back to so much of the immune system function that we really fleshed out was during the AIDS pandemic. So, and I'm sure we're going to learn more about our immune system after this current pandemic that we're all going through, and after every time Ebola breaks out and some of the flu and the flu mutates and changes, we learn a little bit more about our immune system. All right, so we are now jumping into how do we get to that place where our non-specific defenses are working. Maybe they're working at a rate that is okay, but the pathogen is propagating, going through cell cycle division, using our host cells to maybe churn out, like a factory, large numbers of viral proteins, toxins, and molecules, and the rate at which that's happening is faster than maybe our macrophages and our eosinophils and our natural killer cells can find and phagocytose and ingest. All right, and so we need a better way to find the hiding places of the pathogen, a better way to find the cells that are infected and turning out pathogen material, propaganda, toxins, and molecules. And we need a better way to tag it, push in, and try to kill it. Okay, so so that's where we get into our specific defenses. And so for our specific immunity, one of the things we learned, again, is that T helper cells are really, really, really the cornerstone and are critical in bridging the gap between the non-specific defenses working and kind of like the light bulb going off, like, oh, there's a new virus, oh, there's a new bacteria, oh, there's a new fungus, oh, there's a new flute or worm or parasite that's gotten into our system and beginning to destroy the lining of our GI tract or destroy the nasal passageways or eat away at the connective tissue in and around our skin or something. So so when the T helper cell recognizes that, because some pathogens are cells themselves, are entities, they might be multicellular or they might be single cells, we need a way to be able to kill those cells. And we need a way for our specific defenses to, if the pathogen is not a cell, if it is a virus, and it is going to target and utilize our cells, then hijack them to become machines and factories that churn out more viral particles, materials, and toxins, then we need a way to also find and kill and recognize where the toxins are and the molecules and the products are, and we also need a way to find those infected host cells and kill them. Okay, so again, some of our pathogens might be their own cell entities, and they're releasing a lot of toxic materials that need to be, again, small molecules tagged, found, and destroyed. and then the cells destroyed, and then others are small particles that have to get into our cells and then use our cells to disperse more of the particles, pieces, and parts of themselves that can then go and take over different cells. So when we look at how the specific defenses work, the T cell portion of the specific defenses, so the suppressor cells, the cytotoxic T cells, the memory cells, those are going to be our cells to try to find and kill cells. Find and kill bacteria, find and kill parasites, find and kill amoebas that we know are not self. And they're going to also target host cells that are actively building and producing the foreign materials and are putting some of those foreign materials in their membrane-bound proteins to highlight, this is what I'm making. And this is what we are producing through our DNA transcription to RNA, RNA translation to protein, protein post-modificational tweaks to glycoproteins, phosphoproteins, lipoproteins, or just fourth level, twisted twirly proteins. Okay. B cells are going to put together an army of flags that are not about killing anything, but about dispersing more quickly than maybe one or two cells that can eat something can get around and move around. So the antibodies are kind of like our, again, tags to quickly go around and just kind of like swat sticky notes on you're not self, you're not self, you're not self, and or yourself but you're infected, yourself and you're infected, yourself and you're infected. And then again, the antibodies do not kill anything, but once the antibodies tag, then complement, then neutrophils, then macrophages, then natural killer cells, then cytotoxic T cells. Those entities that have the ability to punch holes and kill something can come behind and kill those items. Okay? So antibodies can only bind to entities, molecules, proteins that fit the antigen presentation. And so one thing about the specific defense that is a weakness, and we see this with, like right now, chickenpox. When you're actively sick and when you're actively producing viral proteins, viral molecules, viral toxins, and your cells are actively expressing and producing those viral entities, then you know that you have an outbreak of it and you're killing those cells and you're trying to kill and disposh and destroy the products that those cells are making. You cannot tag and you cannot kill cells that might be infected by a virus but are not actively at that moment expressing the viral DNA, RNA material. So that is why if you ever have something like herpes simplex, the cold and flu, the cold virus on your lips, or genital herpes or whatever, you never cure someone of herpes. All you do is you try to suppress the infected cells from ever expressing that now part of the cell DNA material that the virus injected into it. And when it does end up producing those viral entities because of the way the cell cycle and regulation of what is produced from the DNA is handled, and when those outbreaks happen, then you treat the symptoms and try to treat the cells to produce or kill them. All right. So when you hear about people being cured of HIV, being cured of herpes, being cured of hepatitis, that's really not a right use of the term, because they might always still have cells in their body that have that virus. It's just those cells have not quite yet turned on and started to make those viral entities, particles, to try to, again, expand and release more viral particles and outbreak of a new viral condition, right? And so chickenpox, I have a friend, she actually emailed me, she has shingles, and shingles has broken out in her, and she's really in pain. Well, shingles is you get chickenpox as a kid, and the virus embeds into some of your cells, and the virus is a herpes virus, so it embeds in some of your nervous system and some of your neuroglial cells and things. And she's having an outbreak of those cells now, expressing that herpes chickenpox virus. And this time, instead of it making big red welts that look like pock marks, that, like, you know, I always think of like the bubble wrap. Instead of having bubble wrapping looking red welts all over you, it's causing a streaky red inflamed nervous system, and it itches and it burns and it's painful. All right. And so now the virus is again happening and spreading and infecting new cells, but as it does so, it's presenting in a different way. Okay. And so, yes, could some of the antibodies in the memory cells and the T cytotoxic and T memory cells from before, when the last outbreak was maybe, you know, in her 5, 6 years old, and now she's in her 40s. Could it be that, you know, they're still around? Yes. Could it be that maybe a lot of those memory cells have died off, and so that's why the outbreak is kind of happening and really robustly causing her pain and discomfort? Yes. And if there are some memory cells, they're trying to ramp up and get going again, but we're also depending on new cells to form and attack and hopefully highlight and make new memory cells and new antibodies against the virus that's shown its ugly head again. Okay, so I just wanted to kind of give you that overview that while the specific defenses are very, very good at killing cells, at targeting and sticky notes on top of, you know, pathogens and their products, it has a weakness, and that weakness is very much exploited by the viruses out there. And then in some cases, like tuberculosis, if the bacteria embeds, and they say it makes like a nice like capsule type protective covering of itself, and it embeds in and around your lungs, and it might sit there as this capsule. And that's why after you have a positive TB test, you always have a positive TB test, because every time they put a little bit of tuberculosis, you'll have antibodies show up, but you might still get exposed to tuberculosis bacteria later on, and the next time you can't know if you're newly infected again unless they do a chest x-ray and they look for those capsules, those like little pods of waiting bacteria that's under a protective shell. They're just waiting for immune system to kind of like not sleep well, not eat well, be high stress. Oh my God, coronavirus. And to just then take advantage of that weakened immune system and to expand and propagate and then start eating away at your lung tissue. And then we've all seen Westerns like Tombstone, where we'll then start coughing up blood and we'll start having our lungs deteriorate because the bacteria is attacking and tearing apart the lung tissue. Okay, so let's go into more about specific immunity. All right. So we understand that specific immunity is the third line of defense, and it's dependent upon the first and second lines of defense to come to it with pathogens and show it like, hey, this is inside of us. All right. It does have a little bit of an ability to be randomly like, oh, I bumped into something in the spleen because I'm a lymphocyte, and like, oh my God, this is not supposed to be here, and I haven't been told that this is here yet, or haven't been presented this yet. So now I'm going to ramp up my system because obviously this pathogen is really, really crafty at avoiding my macrophages, my neutrophils, and my other antigen presenting cells. But for the most part, again, your T helper cells are brought a pathogen, are able to go, yes, this is a pathogen. Yes, we need to ramp up defenses. Come over here, B cells. Come over here, T cells. Sensitize to this antigen, and let's find it. Let's target it and kill it. Now, that may, again, T helper cell being presented a pathogen, that might take 3 to 4 days. And then to get cytotoxic T cells up and running, to get antibodies up and running, that might take another 2 to 3 days. So we're looking at the specific defenses might not get their act in gear and their act going until 5, 7, 10 days after you touch the doorknob and then licked your fingers. All right. And we know that there are some pathogens out there that when they get into our system, and because of the heat, because of the fuel availability, because of the warmth, because of the just perfect storm of what we provide for them for living purposes, they can propagate and release toxins and kill cells faster than our system can become aware and mount a defense. And so our immune system might need some help. All right. Now, the immune system, if we lived in medieval times, we would only have the specific immune system of I have to let a virus come in through whatever pathway it comes in. I have to hope that my non-specific defenses find it quickly, present it to my specific defenses, and I get up and running with my antibodies and my cytotoxic T cells before this pathogen kills me. And that example of I naturally get exposed and I wait for everything to take its course is what we're going to call our naturally occurring active immunity. And that is the normal way that before science and technology really got up and running in the 17 and 18 and 1900s, that was the way that most people survived smallpox, polio, tuberculosis. And you can look at the numbers, flu. Not a lot of people survived because a lot of pathogens were going to kill people before their specific defenses could get up and running. Okay. And in many cases, like with babies, it might be that they got so dehydrated and such a high fever that killed them. The process of the inflammation and the fever and the non-specific defenses trying to get the body up and running killed them before the system could get going. Okay, so what we learned with smallpox is that dairy people who got exposed and caught cowpox never got smallpox, never got the human version of smallpox that was grossly disfiguring and like a 20% survival rate. So 80% of the people, so if there were 10 of us in the room, 8 would die and 2 of us that survived would be grossly probably disfigured with the pox. But cowpox people, the dairy maids who had that, they had a little bit of pock. It was like acne pock, like they got a little disfigurement, so not nearly as disfigured, and not many of them died. So that led to, well, there are certain viruses that if we can, again, get a milder form, get a less deadly form, and put it in our system, we could inoculate and potentially not die from the more virulent, more deadly form. All right. And so that kind of led to what eventually becomes vaccines, is that I could like naturally go and try to get myself exposed to cowpox, or once we got lab procedures and technology going, we could take smallpox and the virus and kill it. We could take cowpox, the virus, and kill it, and we could give it to ourselves in an artificial manner and allow that exposure to the non-living, non-propagating pathogen still get our system up and running 7 to 10 days later with antibodies and cytotoxic T cells, and memory cells were created. So that way, if we ever did get exposed to the real deadly, nasty pathogen, our system already has memory cells ready and hopefully ready to ramp up and get that more robust response against the virus, against the pathogen, so we don't get sick and die. And we don't get as bad of an outbreak, and we don't get as much disfigurement. Okay? So again, inoculations and then vaccines were one of the first ways we went, oh, there is a type of way to get the specific immune system up and running faster. But to do so, we need to give it exposure to the pathogen, but we need to give it exposure to the pathogen in a dead or not propagating living form. All right. And so now we do this with lots of things: polio, hepatitis, there's chickenpox vaccine, there's now the Gardasil, and hopefully we can get one up and running against this coronavirus soon. All right, so we'll take a form of the virus that won't be living, and we'll still have the same cellular membrane material, so the same antigens, will present it to people, and their body will then go through the active process of the T helper cell getting exposed to this, the T helper cell going, you're not self, B cells come over, cytotoxic T cells come over, see this, make antibodies, find it and kill it. All right. And that's still an active process where we actively then create antibodies and we create memory cells to the system. Okay. Now, another form of the specific immunity is I don't wait for my T helper cells to get my B cells up and running or my cytotoxic cells up and running. I try to get antibodies into the system faster from a different source. Okay. And the natural way to do this is breast milked from mother to child. All right. So one of the reasons why, if you ever have a kid, they're going to be pretty pushy in certain places, certain hospitals with certain philosophies. They are very pushy about mom needs to breastfeed. Mom needs to try to breastfeed. And again, breastfeeding is not for everybody. Some kids that end up in the NICU, it's not possible because they're attached to so many things. You can't put the baby on the breast to breastfeed. So it's not always possible. So don't ever feel bad if you can't breastfeed. And then once they get people breastfeeding, they like people to breastfeed for, 6 months to a year, even a little longer, because mom's immune system and every pathogen that mom is fighting and creating antibodies against, those antibodies will get into the breast milk and be passed to baby. And so baby then is afforded some protections, because if mom is fighting flu virus and the flu virus gets in baby's mouth and nose, and mom's antibodies against the flu virus are in baby, then those antibodies can tag that flu virus, and then baby's cytotoxic T cells and complement and macrophages and everything can start eating those viral pieces and parts and destroying it before it takes over and tries to kill baby. Right? So that is a piece of I am not waiting the 7 to 10 days for the B cells to upregulate and make a lot of antibodies. I'm going to try to get antibodies in as fast as possible. And we're doing this with the coronavirus right now. You hear about like Tom Hanks and certain people who have had the virus are donating their plasma. Well, what's in plasma? Albumin, globulin proteins. What are the two categories of globulin proteins? Carrier proteins, antibodies. So by donating plasma, we are getting some of the antibodies that were created by Tom Hanks's immune system, by whoever else got the virus at their immune system. And then if we get those antibodies and we, again, put them in a little IV bag and give them to some of our very sick coronavirus patients right now, I am trying to get antibodies into their system that can start putting like sticky notes on the virus and its particles and the cells making viral particles, and I can start trying to get complement and macrophages and neutrophils to start killing those cells as I'm waiting for my T helper cells to get their act in gear and be like, come over, B cell, let's make our own antibodies. All right, so I can kind of like speed up the system of the specific defenses kicking in and those antibodies present tagging things faster. Okay, now, Tom Hanks is obviously not going to share plasma directly with me. So the artificial piece comes into, I have to get those antibodies from other humans, from cell culture or animals that I grow them in large populations, large amounts, harvest them from there, and then I got to like tweak it and either make it a pill, because some of us take antibiotics by pill. I got to tweak it and make it an IV or a solution that I get that way, or tweak it and make it something that a cream or something that I can rub on or put in, okay, or drop in the eye. Okay, so artificial is that it's made, the antibodies are made from a source outside the body. All right. And they are packaged in a way and given to me in a way that's not natural. It's not a natural way to inject it or swallow it, bring it in. All right. And it's passive immunity because I'm getting antibodies in from an outside source that are going to start tagging the virus, tagging the cells infected, tagging the cells that are not self, and killing them with my cytotoxic cells. But the tagging is done from, again, not my originating cells and antibodies. Okay. Now, will eventually, I make my own B cells and antibodies and memory cells? Yes, hopefully. But a lot of people, might not make them fast enough before coronavirus would kill them, or before hepatitis would kill them, or before the HIV virus would turn them to AIDS and kill them. So by giving these antibodies from either mom to baby to help fight the virus, or giving them in a drug form to help people fight the virus or fight the pathogen, we're just trying to help the system again get to that specific immune defense, that third line of defense, a little bit faster than maybe the 7 to 10 days it might naturally take to get our system up and running and activated and making its own antibodies. Okay, so these are, again, when you look at specific defenses, when you look at immunity from our specific defenses, getting our B cells making antibodies, getting our cytotoxic cells up and running, this is kind of dividing it into its 4 categories, all right? So if I am naturally going to expose myself to the pathogen and then naturally going to let my system run the 7 to 10 day course of finding it, tagging it, and killing it, that is what we call, again, naturally occurring active immunity. And people who are anti-vaxxers, so people who say, oh, chickenpox is good for kids to have, this is what they want their kids to do. They want their kids to naturally get exposed to the pathogen, go through the natural active process of letting their system find it, tag it, and produce antibodies and killing cells and memory cells for the future against it. Okay? Now, that doesn't work for everybody. Because again, some of us have compromised immune systems. Some of us have, again, lots of other things going on. Some of us might have organs and we suppress our immune system because we've got a kidney from somebody else and we don't want to reject the kidney, so we take medicine to keep our immune system suppressed. So for those people who waiting for their system to naturally and actively react to pathogens is not a good option, because it's going to probably, the pathogen will kill them before that happens. You might want to, again, vaccinate so their likelihood of getting that pathogen and having to wait for their system to actively respond, and that's artificially induced active immunity. So we still create our own B cells and antibodies and memory cells, but we do so against, the first time, a dead, modified, artificially exposed on purpose pathogen. The other option would be, my immune system is suppressed and my immune system is being blocked or suppressed because I got a kidney that I need to keep. Then again, we can try to, I know that they might be exposed to something, so I start giving them antibiotics outside the body from a different source and not wait for their cells to actively upregulate, so it's artificially induced passive immunity. And then again, the only example we have of naturally acquired passive immunity is breast milk with mom to baby, and then mom in the womb with the baby or fetus. I need you, foot stomp this, I need you to be comfortable with describing these 4 types of specific defenses. How can we get the system to work? What are the 4 mechanisms that specific defenses can be induced to happen, can be kind of ramped up to happen, or assisted? Another big question, and usually we spend a lot of time doing this as group work and letting everybody form arguments, is vaccines. Because, the journal, it was a British journal, and it was a very respected British journal. That's where the whole thing came out about there are certain vaccines that cause autism. And it's been proven false, but it was in a really reputable journal. It was very poorly done science, and the peer review process failed. But because of that, there is now this huge anti-vaccination movement. And it's a big movement in this country because things like whooping cough, chickenpox, yes, chances are most of us healthy people and healthy kids would probably, if we naturally acquired and let our natural adaptive active immunity system work, we would be fine. But not every kid has a working active immune system. Like I said, some kids might have congenital issues, some kids might have cancer going on, they might have things going on that they're fighting against, and their immune system's compromised. And their immune system is then throw this extra little curveball their way, and it's like they're juggling 3 balls. It's the 4th ball that causes the whole system to collapse, and that's death. And it's kind of scary that there's been a whooping cough, there's been a measles, like somebody went to Disney World with measles, and because again, California, Oregon, that spread, and there was like a big measles outbreak. Whooping cough, I think, was big in Chicago not too long ago. And all of those things are preventable childhood diseases that shouldn't kill kids, and people were seeing kids dying. The flu normally kills a lot of people every year, and this year I think a little bit more of a push for the flu vaccine to be taken by people. When I went and got mine at school, 2 of the maintenance guys were there, and one guy said, yeah, I've never gotten this vaccine before. I've never had any reason to get the flu vaccine, but I'm getting it this year because I don't want to up the risk that flu and corona look alike, and then I end up at the hospital and get corona because I went to the hospital with the flu. So those of you that are going to work in healthcare, you don't have a choice. You have to get these vaccines because you're going to work with kids and people that are sick and immunocompromised. Those of us that were in the military, you don't get a choice either. And many times you're walking down the line, they're giving you like your chem mask, they're giving you your chemical and radiation gear, and then you walk to the next person and they have a little gun and they shoot something into your shoulder. And then they go to the next person like, you're going to Asia, here's yellow fever and here's blah, blah, blah. And you don't even have a choice. You're just getting shot in the arm with 2 or 3 things as you're grabbing gear that also could be life-saving equipment. So you don't always necessarily get a choice or a say. Most vaccines, again, the process of making them and the process of producing them has been, once we know what is going to work and what works on the system, it's pretty much a very fleshed out process. What's taking so long right now is what is it that is going to be the most effective at, when we give it to people who haven't had the virus or the disease yet, going to give them the most ramped up immune response, that immune active memory cell making response and antibody making response, that then will effectively, when the virus comes at them, help them kill it faster than it can make them sick. The other big thing about vaccines is they're not a complete cure. Like if I got my flu vaccine, could I still get the flu? Yes. But if I get the flu and I have the vaccine already ramped up my antibodies and my B cells and my memory cells, so I can start targeting and killing cells faster, I should not get as sick. So might I get sick for 2 to 3 days with a little fever and some achy joints? Yes. But if I had the full-on flu, could that have been 2 to 3 days? Not, but 5 to 10 days, and dehydration and high fever in the hospital, and maybe death. Yes. So remember that some vaccines, you may still get a 24 to 48 hour little bug that when you get the real virus, it's still kind of, ramps up a little bit of a response in your body, and it is a little war, and it still gets a little bit of its punch here and there of I'm going to propagate and I'm going to expand, but the good news is that hopefully, because of the vaccine having ramped up the system before the real virus, the real pathogen got into your body, you're able to punch back faster and you're able to kill faster and prevent that sickness from being maybe 2 weeks or 10 days or 5 days, its normal course of destruction. Okay. All right. So be prepared for why vaccines. Again, you should have them or not have them. Herd immunity is a word that you usually hear with vaccines. And the thing is that if we can get out of 100 people, if we can get like, okay, 90% of us are vaccinated against it, and 3 to 4 people can't be vaccinated, the idea is that everybody that's not vaccinated is mostly protected, because if everybody is vaccinated and not going to then get and pass on and propagate the very deadly virus, the people who can't get vaccinated, the people who can't potentially get protection through a vaccine, will be protected, because the odds of them running into someone with the virus that's spitting it out with their talking and their breathing is very, very low. And so what we see in like California, Oregon, and some of those states where they didn't have high vaccine rates for measles, high vaccine rates for whooping cough, is that more kids were getting it because they didn't have the herd immunity effect. And then there was a higher incidence of kids dying from it. With coronavirus, let's say 30% of the population ends up infected with the virus. And again, what we don't know is, can you get it a second time, and how long are your antibodies and your memory cells going to be, workable and keep you protected from getting a second infection. And then let's say we can get a vaccine that gets the other 60% of the population. So if we can get 60% vaccinated, 30% have had it, that leaves, again, 10% of the population that shouldn't get the vaccine, can't get the vaccine, or is immunocompromised. We could potentially be protecting them because 90% of us are vaccinated or previously infected folks. The reason why the flu vaccine has to be given every year is the virus mutates. Ebola vaccines are like non-existent, because Ebola viruses are so, again, mutatable, that we saw this. The guy that came back in that Ebola outbreak to the CDC in Atlanta, his antibodies, they were able to harvest and do a artificially acquired passive immunity for those 2 nurses in Texas. But maybe if it had been 3 or 4 months later, chances are maybe that Ebola virus those nurses got exposed to would have been different from the one the guy in Atlanta got, and he wouldn't have been able to help them with his plasma. And so that's part of the reason why it's hard to make vaccines against viruses that are very, very, very mutatable. I don't know about this coronavirus. If it's super mutatable, then the chances are we won't have a vaccine, and the chances are also that those people that have already gotten it could get it again. Just like the flu, you could get it every year because it changes enough that whatever protections you have for your memory cells and your antibodies against those other antigens from the previous year, they're not effective against the new antigens of this year's virus. Okay, so that's kind of a little bit. So be prepared to talk to me about why we should get vaccines, the importance of them, or be prepared to tell me why we should do away with vaccines. I'm open. This is one of those areas in science where, everybody has their opinion, and technically there's no good or bad answer. Vaccines in some countries are not available, and they make it work with naturally acquired active immunity and trying to have antibodies and artificially induce passive immunity when people get sick. So you can make a case. Either way, in this country, for most schools, they try to make a case towards vaccination and requiring it to help, again, herd immunity induced effects for kids that can't be protected. Okay. All right. So next slide. The next big point we need to make is about T cells, and going into some more detail about, on the membrane of some T cells, there are going to be some certain proteins. And those proteins we've classified as CD4 and CD8. Now, on all the cells of your body, we have antigens on them, proteins, and there is another protein that corresponds to being a binding or a docking station for the CD4 or the CD8 protein. And that protein is called the MHC complex. So it's the multi-histocompatibility complex, MHC, and we have 2 types. We have MHC type 1 and we have MHC type 2. Now, your MHC protein that you make from your DNA is unique to you. It comes from, again, mom and dad. There are some proteins on, let's say, chromosome 1, proteins on chromosome 4, proteins on chromosome 6. And those proteins are made, and it might be 1 chromosome is from your mom, 4 chromosomes from your dad, 6 chromosomes from your mom, and the protein you create with a piece 1 here, piece 2 here, piece 3 here, and then you swirl and twirl them together, that protein, all right, is unique to you, okay? And the closest you have to someone maybe having the same or identical protein is your siblings. And the reason why your siblings is they also got mom and dad genes, and it's possible that, so I have a sister, maybe my sister, all her number 1 chromosomes are from mom like me, all her number 4 chromosomes are from dad like me. We have a difference. 6 for her is dad, 6 for me is mom, but our MHC, if it's 3 proteins, 2/3 of it should be the same as me. Okay, so that's kind of a little very, very simplistic version of this explanation of why MHCs are unique to you and why MHCs are, if you're trying to find someone who's a match, the closest matches may be your siblings. Your parents have the ability to maybe have 50% of the MHC equal to them. And then you start going to blood relatives, might be, again, the next closest, but it starts to be against the normal population, a crapshoot. Okay, so so these MHCs, all right, one of the MHC molecules is on every cell of your body. And what every cell of your body does is when it starts making, let's say I'm going to make a protein that's going to help me break down glucose. So I'm going to make a bunch of proteins known as hexokinase. Hexokinase is one of the first proteins in the glycolysis chain. So I'm going to make a bunch of hexokinase. Well, when I'm making hexokinase at my ribosome and smooth and rough ER, and hexokinase is being swirly twirled in the ER to be the functional protein that then in the cytoplasm takes glucose and turns it into glucose 6-phosphate, I'm going to take 1 or 2 of these hexokinase proteins and I'm going to put it on my MHC. And so when my natural killer cells and my cytotoxic cells and my T helper cells come over and they bind and they're going, oh, MHC, all right, you got the right MHC. What you doing in there? I can show it. Hey, I'm making hexokinase. Okay, hexokinase, that seems kind of normal. All right, fine. Carry on. All right. So what I see is that MHC class 1s are on my cells in my body, and I don't know if I can blow this up, but in both of these pictures, the MHC class 1 is going to be how, when I make DNA into RNA, and I make RNA into some proteins, I can take some of those proteins and put them on my MHC class 1 and put them on my membrane, okay? And when, again, my cytotoxic T cells, my CD, I can't read this, but I think it says CD, or I don't want to tell you wrong. My MHC class 1, I'm going to say it wrong, and I know I'm going to say it wrong. And my CD, it looks like CD8 expressing cell, all right? When they come together, this is how my cytotoxic T cell knows that I am a healthy, normal cell, not invaded by a parasite or a virus, and I'm producing normal proteins, normal cell cycle, cell functional proteins, okay? If I am hijacked by a virus, the way viruses work is they try to inject their genetic material into your DNA. And so they can do that by reverse transcripting messenger RNA into DNA, or they can try to have these little clipping enzymes that cut your DNA, make a spot, and then insert their DNA. Okay? So if they ingest, or if the virus can get its DNA in its cell, and the cell starts making those DNA particles, molecules that are going to bring maybe new viral little complexes together, some of those products that I make from the virus, DNA being transcribed and translated, I'm going to put in my MHC1. And then the cytotoxic T cell with its CD8 is going to bind and go, oh, yourself, but what are you doing in there? What is this? This is an antigen. This is something that's foreign. This is something that should not be inside of you and you should not be making, so we're going to kill you. Okay? So that's how the MHC class 1 happens. MHC class 1's on every cell of your body. So even your T helper cells, even your macrophages, even your cells that are part of the immune system could get infected, and it could be that then they put those infected particles and products they're making because they're hijacked and try to be like, come over here, cytotoxic CD8 expressing cell, and kill me because I'm doing something I shouldn't be doing. Okay? Now, the class 2 MHC is supposed to only be on your antigen presenting cells. So this is supposed to be on your cells like your macrophages, your dendritic cells, and potentially your microphages that are going to eat material at an injury site where there could be antigens, pathogens present. All right. And so the way that works is your MHC class 2 is going to get some of the pieces and parts of the debris that you pick up when you phagocytose cells or phagocytose particles and pieces and parts of cells, and it's going to take part of that and display it on its class 2 molecule, and then it's going to go to the CD4 expressing cell, which is like a T helper cell, and say, hey, look at what I ate. And so the MHC class 2 is part of how our antigen presenting cells present the material they eat, the pathogens they eat, the antigens of the pathogens that they find. All right? All right, so you need to know CD8 and MHC1 usually go together, and MHC2 and CD4 to go together, all right? And so when these dockings happen, that is part of how, again, we think, this is all theory, we think cells go you're self or you're not self, all right? And then they go, what's going on inside you, or what did you eat? If what's going on inside you is not self and something bad, then we can kill you. Or what did you eat? What you ate is not self and not something good. Let's get everybody together and let's start finding and making CD8 expression cells that can help find this antigen and kill it wherever it's, run off, tried to hijack or propagate. Okay? And so that's, again, just more on how the CD4, CD8 cells. Again, it's more complex. We are trying to keep it simple, but that is the gist of MHC class 1 and class 2 are genetically for you, made from your genes. And again, it's a few pieces and parts, so you have to have, again, genetics is unique because you have mom and dad from whatever that sperm and egg mixture, and they help you, again, maybe have pieces and parts of proteins that come together from different places. So you have a MHC that's unique to you. MHC class 1 is on every cell of your body, and that's how, again, every cell can kind of show what it's making inside of it and show, like, I've been hijacked and now I'm making these antigens, so please kill me, toxic T cell, because the cytotoxic T cells tend to be the CD8 expressing cells. And then antigen presenting cells are going to have MHC class 2. And how do they present to the CD4 T helper cells? Well, this is how, because the MHC class 2 and the food antigen particle pieces that were eaten through phagocytosis and digested are brought to the CD8 or CD4 T helper cell, and then the CD4 T helper cell can go, oh my God, bring the B cells over, bring the T cells over. Let's get some CD8 cells made against this, and let's get some antibodies to make against this. Okay? And antibodies, again, they are usually drawn as these Y-shaped proteins, all right? They are going to, again, be 1, 2, 3, 4 little pieces of protein from a, potentially 4 or 5 little spots of DNA, introns and extrons. The part that is always manipulated and made for a specific antibody is up here in this Y region. It's the part that's known as the manipulated piece. The part that's always stable and is the same, so complement knows to bind, is the blue kind of inner middle and the body of the Y. Not the arms, but the body part. And again, your B cell, somehow, when it gets exposed to the antigen from the CD4 T helper cell, it knows, like, okay, here's my intron. All right, I'm going to get rid of you. I'm going to get rid of you. I'm going to keep you, and I'm going to cut this piece of DNA out and bring these two pieces together, and now I have, when I make my DNA to RNA and RNA to protein, I have this unique piece of material that is forevermore going to make this antibody able to find this certain pathogen. All right, and these antibodies are known as immunoglobulins. We usually make them I immunoglobulin, and then they'll get a letter like M, like capital G, like capital A, capital E, and some of that relates back to which little piece of DNA was kept and made to be part of this variable, that's the word, variable segment of the DNA or protein structure. All right, and remember that variable segment is binding and it's tagging the, like a sticky note, saying, hey, I'm putting the sticky note, you're not self, you're not self, you're a particle that doesn't belong, and we depend on other things to come behind and destroy. All right, and then when we put this all together, I'm gonna go grab a doorknob and lick my fingers. All right, pathogen comes in. Most of what's happening is, again, I see ramping up non-specific defenses, or I cut myself. Okay, neutrophils, macrophages, barriers, swelling, non-specific defenses happen. The pathogen that got in because of the finger that I put that was dirty, or the cloth that I put on top of the injury that's dirty, that had some flu, strep, bacteria, all of those little pathogens are trying to get in, and they're trying to, like, you know, bumper cars, like avoid being bumped and squish through the cells and squish through and get into the lymphatic system. And as they're doing that, maybe they're like, ooh, it's warm, ooh, there's some glucose, ooh, there's some fat, and they're going to try to, you know, propagate and mitose and divide and multiply as they spread out. And they're going to make, you know, spitting themselves spit out some toxins that's going to be like, ooh, I want to embed myself right here, so let me make my spot. All right, so when you look at the response, very early on, we see non-specific defenses are happening, you know, 1 day, 2 days, 3 days into the exposure or the initial injury. And it isn't until around that, again, 7 to 10 day window that we start to see cytotoxic cells and antibody numbers come up, and come up in a manner that they're probably tagging and killing faster the pathogen and the particles the pathogen is creating. They're killing and destroying that faster than the pathogen can divide and multiply and create new toxic molecules to spread out. All right, and then again, when we look at getting an exposure to a pathogen for the first time, it's going to take, again, about a week to 2 weeks before our antibodies, our titers, our numbers. So if you try to pull plasma out, it's going to be a week to 2 weeks before I have a lot of antibodies circulating in my plasma, in my lymphatic fluids. And the amount of that circulation is probably, you know, a decent amount, but not super, super high. But I've created memory cells, and those memory cells are sitting in the connective tissues, maybe near that original injury site, in the lymph nodes, and the spleen, and areas maybe near the original entry site. And so if another pathogen ever gets in to the system, and it uses the same mode of entry, like come in with food, coming in with air, those hopefully, again, memory cells in that connective tissue, memory cells in that lymph nodes nearest that area, will then quickly maybe ramp up their defenses when they bump into that pathogen, and get a response the second time that gets the numbers up and running for antibody release faster and higher in the time frame. And so that's, again, why we want, if we can, vaccines against things like cholera, things like polio, things like whooping cough. If we wait for this smaller, longer response, I'm going to be dead right here. But if I have been exposed at that same time point, look at the numbers of antibodies to fight and kill my pathogen. Well, they're up and at them and killing maybe before the pathogen can kill me. Okay. The other beauty of the system is go touch the doorknob, lick your fingers. I didn't give myself exposure to one virus, one bacteria. I probably exposed myself to like 20 pathogens. And my system is fighting all 20 pathogens at the same time. And hopefully a bunch of them are repeat offenders, so I can ramp up those secondary responses and get them dealt with while I wait and maybe have to then let the natural course of naturally acquiring active immunity happen for the 1 or 2 newbies that have somehow, made their way to the doorknob because some stranger came and tried to door-to-door knock on my door and spit on my doorknob or something, whatever. All right, questions. I'm going to stop the recording. That was really, really fast, really, really super, super fast. Hopefully it helps explain, if you've already read or listened to the lecture, how I want you to approach this. Again, I don't need you to tell me genetics. I don't need you to tell me all the nitty-gritty specific details. I need you to, though, know MHC class 1, CD8, MHC class 2, CD4. All right. I need you to know naturally acquired active immunity, naturally acquired passive immunity, artificially acquired active immunity, artificially acquired passive immunity, and the examples. I need you to know B cells, antibodies, the variable in could be a piece of the genetic code that makes it the IgA, the IgE. I don't need you to tell me what IgA is going to fight, what IgE is going to fight, where they're located, just in general, that one antibody is not universally going to work for us. We actually have 5 or 6 groups of antibodies, and then these variable ends are the pieces we manipulate to best work for certain pathogens. Okay. All right. So when Tuesday in lab, those of you coming tomorrow, we're going to talk respiratory anatomy. So look over your nose, leave the mouth for digestion. So nose, pharynx, larynx, trachea, lungs. Get your anatomy of those things and your histology of those things down so we can go through it quickly tomorrow. Thursday, we'll do that again and I'll record it. Wednesday's lecture will be the respiratory system. All right, so we're going to go through some of the big items we have to know about. We're going to stick to the physiology since lab is the anatomy. So we're going to hit ventilation. So I want to hit ventilation and Boyle's law and Dalton's law and the gas laws. Okay.